Hello and welcome to our second session dealing with mitosis and meiosis. In the previous session, we had a look at the cell cycle and the shortest part of the cell cycle, the mitosis. So the phases of the mitosis are the prophase, metaphase, anaphase, and telophase, and this is actually something you should remember. In addition, before the mitosis actually starts, there is a phase called interphase, but technically interphase is not a phase of the mitosis. However, in order to understand mitosis, we need to have a closer look at interphase as well. In the following few slides showing mitosis and certain processes during mitotic cell division, you will see a schematic view of the process on the left hand side, and on the right hand side actually images generated during experiments in a lab. Here DNA is going to be always stained in blue, and the cytoskeleton or microtubules are going to be stained in red. So this will basically give you a better view of the process in a real cell. So, in the interphase, the chromosomal configuration in each of the cell is 2n, so those cells are diploid. What happens here is that cells simply prefer to divide. The genetic material is replicated, so during the interphase, replication occurs. In the prophase, which is the first phase of the mitosis, our chromosomes pair up. And this is quite nicely shown both on the left-hand side, where you can see the chromosomes, and also here on the right-hand side, where you see the DNA staining. Those are all chromosomes. And cytoskeleton is also rearranged. Importantly, during the prophase, replication is already conducted, so the chromosomal configuration is not longer 2n but 4n, because replication already finished and our chromosomes are fully replicated. So the chromosomes are thickened and shortened, become visible for the first time, and the two sister chromatids are joined by a centromere. The centrioles move to the opposite side of the nucleus, and those are the centrioles in the schematic view, and the nucleolus disappears. Moreover, the nuclear membrane, which is surrounding the nucleus in all other phases of the cell cycle, disintegrates. The next phase of the cell cycle or mitosis is the metaphase. And remember, metaphase is the phase where we can actually make chromosomes visible. Chromosomes line up at the equator, and the chromosomal configuration is still 4n. They arrange at the equator of the cell, become attached to the spindle fibers by centromeres. And homologous chromosomes do not associate yet. In the anaphase, the sister chromatids are pulled apart by the microtubules, but the chromosomal configuration is still 4n. And I personally like the image on the right-hand side very much, because you can see quite nicely how those spindles, which are formed by microtubules, are pulling the chromosomes that you can see here apart. So there is clear separation of the sister chromatids. During anaphase, the spindle fibers contract, pulling chromatids to the opposite poles of the cell. In the telophase, the cell pinches in the middle, and you can somehow recognize it in the staining from the lab experiment. The chromosomes uncoil, spindle fiber disintegrates, centrioles replicate, because each of the daughter cells needs to have a full cell of them replicate, and a new nuclear membrane forms. Once this is achieved, we have a process called cytokinesis, where two identical daughter cells are formed, and we are back to diploid configuration. So each of those daughter cells is identical and has a chromosomal configuration of 2n. To close up the summary of the mitosis, I would like to share a nice video with you which shows those processes as a very, very nice animation.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth.
Mitosis is a very important drug target because mitosis can be specifically targeted, for example, to slow down division of cancer cells. And as you might have recognized before, this can be very useful, especially in fast and rapidly dividing types of cancer like glioblastoma. And indeed, there are many drugs that exploit different processes that occur during mitosis. So some molecules are known to stabilize microtubules, thereby preventing mitotic spindle from functioning properly. Other drugs cause microtubule depolymerization, so basically they destroy the spindle fibers. Other drugs can inhibit cell cycle progress and so on. So this is really the important process in targeting cancer cells. And this explains also many side effects of chemotherapy. Because, well, in many patients, you will have low white blood cell count. This is because blood progenitor cells are not longer able to divide, not just cancer cells. Also, many patients experience hair loss because dividing hair follicle are affected, and this should basically highlight to you why those side effects actually occur. With this, I would like to end session number 2, and I'll see you in number 3, focus on meiosis. Thank you very much.